Okay, welcome to the next in our series of bite-sized bioinformatics talks. Today, I'm going to talk to you about a bit of software that we developed actually some time ago, but underwent a fairly major rewrite recently. And this is a program called Nexons, and it's a program for dealing with the problem of quantification and quality control of RNA-seq data performed with nanopore sequencers, okay, which is a relatively new technique still, and one which is offering some advantages over more traditional ways of doing RNA-seq. So just to describe the data and the problem we're aiming to solve here, for many years we've been able to do conventional short-read RNA-seq. So I've shown here a schematic of a single gene with two different splice variants. So on here we've got the two genes and the exons of the first splice variant, and a different one where we have an exon which is excluded from the second variant. In conventional short-read RNA-seq, we are sequencing short fragments of transcripts. So we get transcript fragments which are generally sort of 100 bases or so in length, maybe a little bit more, but nothing approaching the length of the transcript. So when we map the reads that we can produce onto the structure of the annotation, we see something like this. Instead of seeing entire transcripts, we're seeing very short fragments of transcripts. Sometimes those fragments will cross splice boundaries. So the green portions here will be things that cross splice boundaries, but the majority of reads are very local and contained within a single exon. When we come to quantitate this data, it's a very challenging problem to quantitate the data at the level of a transcript. You don't get any context beyond the individual read. So, for example, all of the reads that sit in this final exon are equally compatible with having come from the first or the second isoform on here. Likewise, those in the first or the second exons. It's only really the reads which span a discriminating splice junction, or which exist within an exon that only exists in one splice form, that give a direct signal to discriminate between splice forms. So for all of those reasons, with the complexity that you get in real experiments, it's very difficult to accurately quantitate data at an isoform level with short-read RNA. Now, the more recent advent of long-read RNA-seq, which could come from either nanopore sequencers or things like the Pacific Bioscience sequencers, should aim to solve this problem. So with long-read RNA-seq, the requirement for fragmenting your RNA before sequencing it goes away. So in principle, you can sequence the entire length of each observed transcript to produce data that looks more like this. So now, instead of seeing short fragments of transcripts, you're getting reads which encompass the entire length. So there might be a little bit of difference in exactly where they start or the termination points on here, but all of the splice junctions should be observed, and the problem of attributing reads to splice forms should be completely trivial. If you're sequencing the entire length, then the attribution is just a case of matching up the splicing pattern with the isoforms that are there and doing a simple count of that. So that's the aim and the promise of these sorts of platforms. As with any data type, the data that you actually get sometimes differs somewhat from the theory that you have when you sort of talk about this. So looking at some real short-read RNA-seq, this is an RNA-seq data set around a single gene here. So the gene is at the top, and I've showed all of the splice forms for the annotated transcripts of this gene. This particular gene has a lot of different transcripts. And then down the bottom here, you can see the positions of the mapped reads over this region of the genome. And you can see precisely the problem that you have with short reads, that they are individually very short, and nearly all of these reads are compatible with being present in a multitude of different isoforms on here. Okay, so that illustrates the problem that short read has in distinguishing splice isoforms. Long read should be better, and indeed it is better, but it's not perfect. So here's a visualization of a nanopore RNA-Seq data set over, again, a single gene. And you can see now the reads are much longer and incorporate multiple exons from the transcripts. But in very few cases are we actually seeing complete transcripts running right from the transcription start all the way through to the termination point at the end. There are different methods of preparing long-read libraries. Some are better at capturing the full length of the transcripts that go in. Others are a little more fragmented, but in all of them, there is always a proportion of the reads that don't cover the full length of the transcript. So although you have more ability to discriminate isoforms, it's not a perfect and simple computational job. There is still a degree of inference to be done. And this is the problem that we're trying to address today with nexons, is finding ways to quantitate and evaluate the quality of these sorts of long-read mapped data sets. Now, one of the biggest problems with this type of data is the complexity of the transcriptome. And we are going to use the annotated transcriptome from a genome assembly to guide the annotations and the counts that we're going to generate. And if we look at an example of a transcript list for a single gene in the human genome in the Ensembl database, this is maybe a slightly extreme example, but it's not at all unusual. What you find is that there are a huge number of transcripts annotated for many genes in the human or mouse or any of the sort of high eukaryotic genomes. So down here, we can see there must be sort of 20, 30 different isoforms listed. One of the things to realize about gene builds in these genomes is that they aim to be as complete as possible. So although there are this many isoforms listed, in practice, only possibly quite a small subset of all of the annotated isoforms here are ever going to exist at any meaningful level in real biological samples. The annotations in here aim to give us some clues as to which of these transcripts we should trust more or less. So you can see in the column over here where it says biotype that some of these are annotated as conventional protein coding transcripts, but if I just blow up part of that, you can see some of them are annotated as misspliced forms. So we have isoforms that should be subject to nonsense-mediated decay. This is a surveillance mechanism that says that for protein coding genes, unless your coding region extends into the last exon, so if the ribosome doesn't actually make it through all the way into the last exon, then there is a surveillance mechanism that identifies and degrades those transcripts very quickly. So even if they're created, they should be destroyed very quickly. There are also examples of misspliced forms. So down here we have some which appear to be a retained intron. And if you take the largest sort of possible set of things that have ever been observed in the human genome, then mistakes in splicing happen and they will be captured. So some of these will be isoforms with very little to no evidence that they exist in a meaningful manner in real tissues. The thing that we can use to help this is in this final column here on Ensembl, we have a series of flags. And the flags are different metrics which are used by Ensembl to try and indicate the reliability or the trustworthiness of the gene model that has been described here. Now, there are several different schemes that Ensembl uses for this, but for the purposes of the tool that we're describing today, we're going to focus on just one of them. And this is what's called the transcript support level. So you'll see on these isoforms here that you have a little tag that says TSL1, TSL3, TSL2, whatever. You get a numeric value for this. So the TSL value, the transcript support level value, gives an indication for how much real experimental evidence exists to support the isoform structure which is being described. It's a numeric scale which starts at 1, which is the highest and most reliable value, and goes down to 5. Well, it goes down to NA where there really is no evidence at all. But basically the lower the value, the more reliable the transcript. So ideally what we would want to see is a transcript support level 1, which is that all of the splicing decisions in that isoform are supported by good experimental evidence. For TSL2, you have evidence for all of the isoforms, but at least the one of the ESTs that supports that, so the cDNA sequences, is flagged as suspect in some way. TSL3 says, well, there's only one sequence that's ever been deposited that supports this, so it could be right or it could be just an aberrant form. 4 is there's only one sequence and it's suspect. And then 5 is there isn't a complete sequence deposited that supports this structure. It's pieced together from other things. So you can see that we have this sort of hierarchy. And this is going to get used as part of our quantitation. The tool we're going to describe is called Nexons, and it's available from the URL up here in the GitHub repository. It's a pretty straightforward tool to use. So it starts from 2 pieces of data. It starts from an aligned BAM file where your long-read data has been aligned against a reference genome. Okay, it doesn't really matter for the purposes of the tool which of the different RNA, long-read RNA aligners has been used. It should be compatible with any of them. The other thing you provide then is a GTF file. So a GTF file describes the genes and transcripts in a gene annotation for the same genome that the BAM file was aligned against. Because of the details of some of the filtering that the tool does, although you can use any GTF file, it's really been developed with some of the conventions used in the Ensemble database. So ideally, using a GTF file taken out of Ensemble is going to give you the best experience with this. The output from Nexons is going to be twofold. So it will firstly produce count tables, so simple quantitations of the number of times each transcript or gene was observed in each of the BAM files that is analyzed. You can analyze several at once for all of the transcripts or genes in the genome. And it actually produces 3 separate count tables depending on the reliability of the attribution it's able to produce. And we'll talk about this in a second. But basically you get 3 count tables, 1 for full-length transcript matches, 1 for partial but unique transcript matches, and the 3rd for unique gene-level matches. And those you can plug into a conventional RNA-Seq statistical analysis using tools like DE-Seq or EdgeR, and they would feed into that sort of conventional analysis, and you can replicate what you would have previously done for short-read data. Alongside the count tables, you also get an HTML QC report, which picks out some of the key metrics from the alignment or from the mapping of the alignments to the annotations and some of the properties of the data along the way. Nexons is a command line application. It's written in Python. And the usage is fairly straightforward. This is a slightly cut down version of the options that are on here. But the main options on here are the ones that we would want to look at. So the things that you have to provide is a GTF file of gene annotations and 1 or more BAM files of data to quantitate. Additional things that we can look at, we talked about this transcript support level value. We can filter the transcripts that we're going to analyze based on their transcript support level, and we can set the maximum transcript support level that we want to use when we're quantitating our data. So if we want to be as stringent as possible, then we could say max TSL is equal to 1, which means only transcripts with the highest level of evidence and the most reliable transcripts are retained for the quantitation. If you don't put a value in there at all, then all transcripts are retained, and you can pick a filter level that's there. Outbase just gives you the base name for the output files. And then we have some flexibility parameters. One of the downsides of doing long read sequencing is that the accuracy of base calling, and particularly the accuracy with which insertions and deletions appear in your data, is not as high as it would be for short read sequencing. So we have 2 types of flexibility in here. We have a flexibility which allows for a certain degree of mismatch in the positions of splice donor and splice acceptor sites in your reads compared to the ends of the annotated exons for internal exons. And then we have a separate end flex value, which is the flexibility of matching the very start of the transcript and the very end of the transcript with the start and end of the annotated transcripts in the data. Okay, because that can be more variable than the internal mismatches. I'll show an illustration of this in a second. Finally, we can specify the directionality of the library. So there are different kits for producing these libraries, and some of those retain the directionality of the RNA. So you can choose to say that your incoming library has no directionality, so reads mapping to the top or bottom strand are both viable for counting against any gene. Or you can have same strand specific libraries, which means that the direction of the reads matches exactly the direction of the annotations, or opposing strand, which means that the direction of the reads is going to be the opposite to the direction of the annotation. So that's just going to be a property of the library type that you've created. This would be a typical example of a command line to run this. We'd run nexons.py, we'd set the maximum transcript support level, in this case we're setting it to 1, the most stringent. We're going to say that the directionality of the library is its same strand specific, and then we will provide the BAM files that we wanted to quantitate. That's it, it'll go off and do the work on that basis. Let's look at what the program is going to do internally. So here I again have a schematic for a single gene that's being quantitated, and now we have 3 different isoforms for that gene that appear in the GTF file and the gene build. This time I've included not only the structure of the transcripts, but I've included the annotated transcript support level. So these first 2 isoforms have TSL1, so they're very well supported and very well documented transcript. And the last one is TSL5. So it's there in the gene build, but there is very little evidence that this is a real transcript. So this might be a slightly sort of sketchy isoform that has made it into the data set. We're going to look at the mapping of reads to the isoform structure. So if I put my first read out of the data up, this read looks like this. I said that we had some flexibility values. So when we're matching the aligned reads against the annotations, we have 2 types of flexibility. We have positions like this, where we have the end of an internal exon that may not match exactly against the spliced positions of the aligned read. So that will be the standard flex value. So we might say, well, you can have like 5 bases either way. So you've got to be close to that position, but you don't have to be exactly on it. And then at the start and the end of the transcript, we have a more permissive end flex value that says how close the end of the read needs to be to the end of the annotated transcript, or the start of the read needs to be to the start of the annotated transcripts. These values can be larger either because you have a little bit of degradation at the ends, or particularly because the annotation of the termination points of transcripts is not hugely accurate and actually is just more variable in different tissues. So we give a greater degree of flexibility for that. What we can see here is that there is a very clear match between this splice form in the data and the first isoform in the annotations, and that this runs right from the start to the end. So this would be a full match of this read against that isoform. If we look at another read in here, we can see that we have a shorter read, which starts in this second exon, and we observe the splice through to the final exon, and we see a single match over here. We are potentially matching only one isoform in our collection of annotations on here. Because this splice goes from this exon through to here, only this isoform has that, and we're not matching all of that. So we're not matching this first thing. We start in the middle of this exon and we finish before the end of this, but it's only compatible with that isoform. So this would be a partial match, but it would be a unique partial match because it only matches that one isoform. Let's look at a final example on here. So now we have a read which starts pretty much at the start of all of the isoforms. We observe this first splice junction, and then we observe this second splice junction into this exon, but then the read terminates in the middle of that exon. This read is compatible with both the first isoform and the third isoform in here, and we don't have enough context to distinguish between the two. If it had extended down to this portion, then we'd have been able to assign it uniquely to one or the other. But as it stands, this is just going to be a gene level annotation because we can be sure that it comes from this gene, but it could have come from either the first or the third isoform in here. At the moment, we are incorporating all of the isoforms in here, and you'd think that would be a good thing to do because more data is better, but it's not always the case. So in this case, we can only quantitate this to gene level, but if we had applied a transcript support level filter and we'd said, well, let's only include transcript support levels 1 and 2, so the higher end of this isoform would have been excluded. And if we exclude that isoform, that would turn this final match into a unique match. So those are the three classifications we can have, depending on the extent of the reads and the structures of the isoforms that we retain in the analysis. We can either take each read and map it as a full and complete match to a single isoform, as a partial but unique match to one isoform, or as a match which is compatible with the collection of isoforms, but we can't tie down to a single one, in which case it's just a gene level match, okay? And we count those matches up and we report all of those matches in separate count tables. So in your downstream analysis, you can choose the degree of resolution with which you want to work. Nexons, when it runs, generates some QC parameters and generates a single HTML output file along with a text file with parsable versions of this data if you want to incorporate that into anything else. But I'm going to show you the information that's put together in the QC report. So at the top of the report, we just have the setup of the analysis. So we have the file that was analyzed and the version of the program and the options that were used when it was run. Okay, so just so we know what was done. We then have some visualizations of different properties. We firstly have the breakdown of the data that existed in the BAM file. So within that BAM file, we divide the reads that we have into those which didn't align to the genome. So the red portion on here are reads that were in the BAM file, but with no alignment to the reference genome. We have reads that are a primary alignment, so they are the best alignment for that read. And then we have secondary alignments, which are another alignment or possibly several other alignments, which might be reported for a read where there was a better primary alignment reported. Depending on the program that you've used to do your alignment, you may not see the unaligned reads in here. They may not be produced in the BAM file at all, and you may not have any secondary alignments. Okay, again, it depends on the program that you're using and the option that's set in it, but this sort of gives you a breakdown of everything that was in the BAM file. Ultimately, it's only the primary alignments that are actually taken forward for quantitation. We then have a breakdown of the degree of precision with which we were able to map the reads to genes or transcripts. So from the set of primary alignments, we can then break these down into those that had a gene level alignment. So the green portion on here is things that could be attributed to a gene. So that means either it was a gene level alignment directly, or it was a partial unique match or a full length match, and we can still record those against the correct gene. So in this case, you can see the vast majority of reads were able to be aligned to a gene. Then you've got those that are aligned to a specific uh, transcript, but were not full length, so they were a partial but unique match to a transcript. The red ones are the ones that were full length matches to a transcript. And then the purple ones are matches that were compatible with more than one gene. So if you were compatible with overlapping more than one gene and you could have been doing that, that's not reported and not counted. So generally, well, you'll always see the gene level quantitations will be the highest, and you would hope that most reads were assigned to a gene. And then you can see the breakdown of how many times you can uniquely but partially assign to a single transcript or get a full length match to a unique transcript. Again, depending on the completeness of your data, you will see different proportions coming up in there. We talked about the directionality of alignments, and if you've run this in a non-directional mode, it will tell you the proportion of reads that matched to the same strand as the gene that they were aligning to or the opposing strand. You would normally expect that the values for this will either be that they will either pretty much all be on the same strand or all on the opposing strand, or about half and half on same and opposing. So this is a non-directional library. They're about half and half. Anything else would be unusual and strange, but it will illustrate that for you. We get some other metrics in here around the data. So we get a graph showing us the range of read lengths that are in here. So we can see that in this data set, although it's a long read data set, and we're getting reads above 2 KB in here, the majority of reads are actually sort of only a couple of hundred through to a few hundred bases in length. So this is not a particularly complete library. It's a little bit fragmented. Again, other libraries may give longer lengths. They will in the end reflect the lengths of transcripts in your data as well. So it doesn't really tell you how complete they are. It's just showing what's reported. And we can also see the transcript coverage. So if you're concerned about having a 5' or a 3' bias in your library, this will show you the percentile through the gene on here and what proportion of the gene is covered by transcripts that are aligned. So we expect to see a bit of a drop off right at the start and right at the ends where the degradation happens, but you'll also see if you get a strong bias to one end or the other. The final metric that we see is how well the reads that we got aligned against the isoforms are based on the flexibility that we allowed. So we talked about two types of flexibility around the start and end points of interior exons, and flexibility around the ends of the gene at the start and ends of the annotated transcripts. And we can summarize that. And in this case, you can see that even though we know that there are inaccuracies in nanopore data, they've really got a lot better over recent years. And although we allowed some flexibility, the vast majority of our inner exon matches matched exactly on the base that they were supposed to in here. Okay, so very few actually showed any deviation in the inner exon position. Even on the ends of the reads, we put this on a larger scale, but you can see the vast majority in here are within, I don't know, maybe 20 bases or so of the annotated start and end positions. So you can see how the accuracy of your gene build and the completeness of your transcripts match up. Finally, we have count tables, and just to show what these look like, you get 3 count tables out of this, one recorded at the gene level, one recorded at partial matches to transcripts, and one as unique matches, unique full-length matches to transcripts. And the tables themselves are very simple count-based tables. We've got the annotations in here. This is one of the transcript tables, so you've got the gene ID. Oh, sorry, this is the gene level analysis, not the transcript. So you have the gene ID, the gene name, the position within the genome, the directionality, and then the raw counts for how many times that gene was observed in the data. Transcript level would be exactly the same as this, but would have an additional column for transcript ID and transcript name. Okay, but other than that, it would be the same. Nexons is available now, and we've been using this for real work in our data. This is the URL from which you can download it. It's a small Python package, very easy to install, and we are keen to get feedback from people who are using this. So we would encourage you to go and give this a try, report back any problems to us. If you have suggestions for how it can be improved, we'd be very keen to hear that as well. And I hope you find some use for this in your own research. Thank you.